Hello, everyone. Welcome back to Science in the Age of COVID-19 seminar series. As always, we're delighted to have you all here with us today. We'll continue our discussion on the therapeutic efforts towards SARS-CoV-2. Today, we'll hear about the convalescent plasma therapy, the idea of using sera of patients who have recovered from SARS-CoV-2 infection as a treatment option. It's a pleasure to welcome Dr. Arturo Casadevall, who is the chair of the Department of Molecular Microbiology, and Immunology at the Johns Hopkins Bloomberg School of Public Health, and leads the COVID-19 Convalescent Plasma Project. Dr. Casadevall, on behalf of all the viewers here, we thank you for taking time off of your work and joining us today. It's a pleasure to host you.
Thank you. Can you hear me? Yeah. So thank you, Sarada, and thank you, Lauren, for inviting me. So what I'm going to be telling you about the efforts in the United States and the world in developing convalescent plasma for the therapy of COVID-19. So I think the first thing is to define what is convalescent plasma. Convalescent plasma is the liquid part of the blood once you recover. So today, your blood, your red cells are floating in liquid. That liquid is plasma. When people recover from an infectious disease, they have antibodies to that infectious disease. And these, in the case of COVID-19, that plasma has antibodies that neutralize SARS-CoV-2. So the way to think about it is that you take the red cells, you spin them down, the blood, and the yellow material is what is used. Now, in the old literature, you hear the word serum. Today, you hear the word plasma. How are they different? Serum, it means that you let the blood clot and you spin down the liquid. Plasma is done in a plasmapheresis machine. Today, we use plasma. Plasma has all the coagulation components. Serum is coagulation depleted. So in the old literature, you will see the word serum. Today we use plasma. So you can take the material and you could give it to people to prevent or treat infection. This is old stuff. It goes back to the discovery of antibodies in 1891 by these two individuals, Emil von Behring and Shiva Saburo Kitasato, who it was known at the time that you could immunize an animal and make them resistant to reinfection with an antigen. But the major discovery was that you could transfer immunity by transferring the liquid part, in this case, immune sera. And this was one of those landmark discoveries in immunology that had far-reaching effects in both medicine and in the development of the discipline of immunology. The first Nobel Prize is given to Emil von Behring. They left out Kitasato, thus making the first Nobel Prize controversial, a tradition that is maintained to this day for every Nobel Prize. But you could see that within a couple of years of that, it was already used for the treatment of diphtheria. The treatment of diphtheria was a very dramatic advance. Diphtheria, this is a painting by Goya, the Lazarillo de Tormes in 1819. You see a boy dying of diphtheria with whoever the physician or the father trying to maintain the mouth open. Basically, a membrane develops in the back of the throat and children asphyxiate in front of their parents. It is a horror that I have never seen, and I'm glad because today we have vaccines in the form of diphtheria toxoid. I want to read to you something from that time because I think it gives you a sense on how this was a miracle cure. I found the boy very ill, the whole back of the throat being like a white velvet. That's the membrane. I had never used the new remedy before, which is serum. But determined to try to save the boy's life, I injected a small quantity under the skin of the stomach and watched the throat. I can only compare the marvelous result to the disappearance of snow beneath a hot sun. After the second dose, every trace of the membrane disappeared and the boys soon recovered. I think that even that story, 130 years later, is quite dramatic. So serum was then developed for the treatment of many infectious diseases. Many companies, pharmaceutical companies, got to start making serum. Sera could be used for the treatment of, for example, pneumococcal pneumonia, meningococcal meningitis, and it dropped mortality by about 50% in each case. Serum was the first time that physicians could treat an infectious disease. The antibody sources came from animals if you had the organism. If you could grow the bacteria, you could immunize a horse, a rabbit, or a chicken. That's preferable because you had a steady supply of antibody. And so animal serum was used primarily for bacterial diseases. But the problem was they didn't have virology yet, so they could not harvest virus to make an animal serum. So they had to rely on convalescent sera. So for diseases such as polio, measles, and others, they took in people who had recovered, and they took their serum and they transferred it to children and to people who were either at risk or had the disease. They had no knowledge of blood-associated diseases at the time. So antibody therapy, the heyday is between 1900 and 1940. And then sulfonamide and penicillin are developed, and the field of infectious disease walks away from antibody therapy. There are some areas, you can see those listed there, some niche areas where antibody continues to be used. But today, if you walk into a hospital, most antibodies are used in oncology and rheumatology. This is a problem that hopefully COVID-19 will hope to change, because what you're about to hear is the largest use of plasma in the history of humanity that I'm aware of. So convalescent serum or plasma was used in many epidemics in the past. It was used in the influenza epidemic of 1918. It was used in many outbreaks, and I'm going to be telling you about this case in a little while. And as late as 1946, it was used for treating mumps because it would prevent orchitis or inflammation of the testicles that can result in sterility. When patients respond, the response was rapid. You saw the story with the kid. Well, here you see a soldier in 1918 who's getting into trouble with influenza. The respiratory rate is rising very rapidly, and this individual will not be able to maintain it. So they give him serum taken from another soldier, and rapidly it effervesces and the respiratory rate returns. So that was an anecdote, but did it work? Well, people went back at the early this century and reanalyzed all that data and concluded that yes, it worked. That a meta-analysis showed that in generally, it fit about 20%, it reduced mortality by about 20%. So convalescent seroplasma was used in 1918. It was used in many outbreaks in the United States and throughout the world. In the 1970s, in Argentine hemorrhagic fever, randomized controlled trials showed that it reduced mortality by about 90%. In SARS coronavirus in 2003, it reduced mortality by about 60%. It was used in the H1N1 epidemic. It's been used in the MERS respiratory syndrome and in the outbreaks in West Africa. I'll tell you more about these. And now in 2020 for COVID. So what are the lessons from history? Antibody is effective for the therapy of infectious diseases. Efficacy depends on timing. The earlier, the better. This is not like you could give the drugs late in the course. Antibodies work through the immune system, and you really need to have it early to neutralize the inoculum. Efficacy depended on the amount. A certain amount was needed. Efficacy, these therapies were well tolerated. Recovery was often rapid, something that has been forgotten today. For pneumococcal pneumonia, recovery was seen within hours. Today, we usually have to wait several days with antimicrobials. But it was very different. There were big differences in whether on the easiness of making the serum. For example, it was easy to make them to diphtheria toxin. That's a simple antibody toxin neutralization. More difficult to make it for pneumococcal pneumonia, and most difficult to make it for tuberculosis. So different pathogens, different pathogenic strategies, different antigenic types gave very different problems. And antibody therapies were largely abandoned in the 1940s. Why? Well, here came penicillin. And penicillin was a lot easier to use than serum. You didn't have to type the organism. You could just basically give a drug as a pill. And this was enormously easier, and the efficacy, by the way, was never really shown to be very different. And for plasma from humans, this led to the association with hepatitis. And you can see one of the early reports, hepatitis as the prophylactic serum. This was an orphanage or so where children were given plasma and resulted in several cases of hepatitis that were to death. And I would like to point out that we have progressed quite a bit in the last 80 years. We don't call people mental defectives. They were treated with less than 5 cc's of serum. And some of these cases develop rapidly fulminant hepatitis a couple of months after the infection. So there is 2 things I want to point out to you here. Antimicrobial therapy was accepted and is used today without a single randomized clinical trial. That becomes important in some of the debate that you may hear a bit later. And the second thing is an anecdote. Today, if I basically said to you that 3 patients recovered, you would say it's anecdotal. But if I said to you 3 patients died, oh my God, that's very different. We judge the value of information very differently. And in this case, these anecdotes will eventually lead to the discovery of blood-borne viruses, the hepatitis viruses. So there are 3 principles that you could say from the era of serum therapy. These were never written down, incidentally. They were common knowledge. And one of the things I'm trying to do is to write them down and publish them because people have forgotten about them. You have the specificity principle. Effective antibodies must contain antibodies specific to the target. You have the quantitative principle, that you must have sufficient antibody to mediate protection. And you have the temporal principle, that antibody preparations are most effective when given prophylactically or early in the course of disease. Here is data from 1913 in the Journal of Experimental Medicine, and you could see that mortality significantly less if you give it in the first 3 days than if you give it after 7 days. So what are the efficacy of convalescent plasma in past epidemics? Well, influenza reduced mortality about 20%. In the Argentine hemorrhagic fever, 93%. For SARS-1, about 73%. For the influenza, 2009, minus 60% or so. And a meta-analysis that I'm going to show you suggests that it's about the same, maybe 50 to 60%. However, there have been 2 major studies that have not shown efficacy. One of them was a randomized controlled trial in West Africa, in which it did show that those who received plasma were much less likely to die than those that didn't. But then when they corrected for age, all these things, the effect was reduced to 8%. They lost power and became non-significant. And then there was a seasonal flu study a couple of years ago that showed no efficacy. So can we do a post-mortem on those 2 trials and see why they may have been negative? Well, in the West African, there is a problem with the quantitative principle. This was done in an under-resourced area. They had no capacity to measure the amount of antibody. They took people who thought they had recovered from Ebola, and they did not measure antibody when it was used. And then a subsequent study that showed that many of the samples had no antibody whatsoever. And in this study, there were problems with the temporal principle. 44% of the patients were already in the unit with advanced disease. And we know that antibody doesn't work very well late in it. And there may have been a problem with the quantitative principle because they consider high titer serum to be 1 to 80. And when you think about the amount of antibody, you may have needed a lot more. So convalescent plasma, summary, is a storied history dating back more than a century. The problem of making antibodies therapies work catalyzed tremendous progress in immunology in the 20th century. 7 Nobel Prizes were given in immunology related to antibody discoveries. And this was the major cutting edge type of work, I would say, in the first 50 years of the 20th century. It had been used emergently in many epidemics and outbreaks, and that is a problem. Because when you use anything emergently, you often don't have the time to do randomized controlled trials, the type of trials that will give you the kind of answers that we like to have in medicine. It can be rapidly deployed. I'm about to tell you that in the United States, in March 1st, there was no capacity for doing this. And by August 1st, 50,000 people have been treated. This is an example of rapid deployment. It is relatively cheap. A unit of plasma, which is a dose they use to treat, costs about $300. And it is easily deployable in underserved regions where they're not going to be able to afford antivirals or monoclonal antibodies. So let's go to COVID-19. So I think you can tell that I'm very interested in history, and I read a lot of history in my field. And I urge everybody to become experts in the history of their fields. And when this was by January, I'm an infectious disease doctor, I knew this was not containable. When I heard they locked down Wuhan, this was just not going to be containable. And I began to pay very careful attention to what was being said by the health authorities. People were talking about vaccines, antivirals, monoclonals, but you never heard the word plasma. And I began to wonder what's going on here. Maybe these people just don't know about this. So the question was how to get the word out. Well, I'm an editor-in-chief of a journal, deputy editor of another journal. I could have written an op-ed, an editorial, but you know what? That's not going to work. I decided that to try to get the work out was to write an op-ed that I could put in a major paper, and then I could take the link and send it to anybody. Why an op-ed? Because people will respond to it. And also because the paper will rewrite it for you, so it's a lot better than anything I can write. And I tried the New York Times, they didn't want it. I tried the Washington Post, they didn't want it. But fortunately, I sent it to the Wall Street Journal in one of those late days in February when the market tanked. And they took it, and they rewrote it, and they published it. And this was a big break. Let me tell you what I wrote about. I wrote about this episode in 1935 in a school in Pennsylvania. This individual, Roswell Gallagher, was the doctor of the Hill School in Pottstown, Pennsylvania. And a kid came down with measles, severe measles. And there was a breakdown in the quarantine such that as many as 66 kids who had not had measles had been exposed. So what he decided to do was to take the blood from the kid who recovered and use it to prevent disease in the others. 10 cc's was considered adequate, and what he found was that they expected 25 cases based on the penetrance, on the contagion of this disease. They only got 3 cases, all that were attenuated. And it's the kind of thing that you could write about because you can connect with the human experience here. And I knew that it was kind of important in doing so. So the next day I took the op-ed and I sent it to all my friends. And I basically said, we got to try to do something about this. I tried to reach government and all that. But you know what? Ultimately, it was my friends who answered me. And here's I reached out to Michael Joyner. He's a anesthesiologist at Mayo, and he's going to become a very important part of this story. And he answered right away, and he said, wow, I never heard about this. Let's think about it. And what he did was he alerted Mayo. Actually, I think what he said to me was, it sounds good, it sounds promising. And then he went on to alert Mayo. And this was very important because Mayo began making preparations very early on, and they were in a position to, in fact, put together what is known as the Extended Access Protocol that has become the major program for convalescent plasma. So my initial contacts, Lysiane Porofsky, a collaborator in New York that I've known for many, many years, she brought in New York City. Nigel Paneth, an epidemiologist at Michigan State University. Michael Joyner that I mentioned to you. Shmuel Shoham here, a clinician in Hopkins, immediately began to write the protocols. And then Michael Joyner recruited Jeff Henderson and Brenda Grossman. So this was a coalition of friends. We began to try to get this set up pretty much on our own. And within a few weeks, we had organized the National Convalescent Plasma Project. These are the people that were involved. Amazon built the website for us. We had no money. And around this time let me just give you a sense of what was happening during this time. Is March 2020. So here's the Wall Street op-ed. I sent it to all my friends, begin discussions at Hopkins, assemble a team at Hopkins. And then we realized that you can't write protocols with an op-ed. You got to have all your references. You got to have all these things that you need. So Lysiane Porofsky and I wrote a paper that I'll tell you about in a minute. In 4 days, the Journal of Clinical Investigation reviewed it in 1 day and put it in the website the next. And that's really important because now people can get the paper, can get the references, and all the protocols that need to be written. So then we organized this group, and this is the time in which I was telling people that I had to take my email off the website, because by now the media was covering this stuff, and I began to get a voluminous amount of emails that I could not possibly keep up. So my admin changed my email address to convalescent plasma. That's still on the website. And then I had people in the department basically screen that site to look for important emails, and then they would forward them to me. I simply could, when you're dealing with thousands of emails a day, there is just not enough time to do so. So I sought Al Sommer's advice. Al Sommer had been a prior dean here, and he basically contacted the Bloomberg philanthropies, and they were able to secure $3 million very rapidly. The only money that we were to get for many, many months. However, the FDA moved rapidly and allowed compassionate use by March 24th. And by March 27th, the first patients were treated in the United States. So this is what I call COVID time. Things are compressed, things happen very, very rapidly. What was happening in the background was that the editorial was written, and then there was a lot of media associated with that editorial. And consequently, and this is why I began to get thousands of emails, was because many physicians wanted to do this. They had nothing to give their patients, so the FDA began to receive overwhelming numbers of requests, and they created the extended access protocol and contracted the Mayo Clinic to run it. And Mike Joyner will then become the PI of the extended access protocol. We all work very, very closely in all of this, but it gives you a sense how things happen behind the scenes. These are the two publications. The first one is what I call the manifesto. Notice that I'm still living in the past. I'm calling it Zira instead of plasma. But then we realize that, this is really complicated. You can't just write, you have to write a how to doing it. So by then, a whole set of people had joined the project, including these two superb transfusion medicine physicians, Evan Block and Aaron Tobian. So this became, we wrote a second within a couple of weeks, a how-to guide. And this paper will tell you, if you want to do this, this is what you got to do. This is the screening and all that. And these two papers now become foundational for setting up this effort. By the way, we put everything in the website, and we gave everything we had to everyone in the world. The world began to download both papers, protocols, insights, everything. We felt that everything had to be completely open, and you can go to the website today and you can download all this stuff. One of the problems that we have in setting this up was that we needed a place to put the website. And to the credit, Nigel Paneth went to the dean of Michigan State University, and the dean basically says, we are in a crisis, put up the website. If we get sued, we'll deal with it later. Anyway, that's the story that I've been told. So the FDA moved rapidly, and we were very lucky to have superb colleagues at the FDA who are hematologists who realized that this could be helpful. And you can see that within weeks, expanded access was provided, and then this program was run at the Mayo Clinic. What this meant was that any doctor in the United States could register a patient if they went to this website and could get permission to treat plasma. And initially, we thought that this may be given, maybe a few thousands. Little did we know that the plasma growth will grow tremendously, such that we're probably approaching, we are definitely past 60,000. We're probably approaching 80 to 90,000 people treated in the United States. The reason I don't have any more final numbers is because the rise has been so great that not all the paperwork has been completed. So we don't have, a lot of people have been registered and transfused, but the doctors have not yet filled out all, especially in the search areas in the South and Southwest. Around this time, China begins to suggest that convalescent plasma is useful. This is very important. These are not clinical trials. These are just case series. They show, provide encouragement, because one of the things they report is that it functions as an antiviral, reducing viral titers. Italy begins to use convalescent plasma. Even though they don't put the information out scientifically, it's in the newspapers. So we're getting people translating Italian newspapers to find out what's going on. But as you can tell from my accent, I'm Cuban originally, and I speak Spanish, so I can tell that plasma funciona. That sounded pretty good. And eventually, their data is put as a preprint, which is again very encouraging. They're reducing mortality, and that paper has now been published. So, however, from a scientific point of view, there began to be a crisis around this time, which I call the antibody-dependent enhancement crisis. And the problem was as follows. If you went into the literature, you would have learned that there is a phenomenon known as antibody-dependent enhancement with coronaviruses. And there are papers like this that were published a year earlier that if you give antibody to a coronavirus, you can make the disease worse. So there was a concern, and many well-known virologists and immunologists wrote me about this. Now, I knew about the history of plasma, and I simply could not believe that if you recover from the disease and you're well, and you take some of that plasma and you give it to somebody who's sick, that you're going to make them ill. But anyway, it was a concern. So the question was how to deal with it. Well, my approach was to read deeply into the literature and to basically contact a lot of these people and talk to them about it. By also reassuring what's information from China, that convalescent plasma had been used in 2003 without any problems, and the news reports from China were favorable. The Italians were not reporting any problems. The Chinese were not reporting any problems. So I thought about, and I went to the wisdom of antiquity, and the wisdom of antiquity can be summarized in these two things down here. One of them is audentes fortuna jubat, which has been attributed to many, but it means basically fortune favors the bold. But you could also find a warning from Democritus that boldness is the beginning of action, but fortune controls how it ends. So be careful. But we felt that the overwhelming, in particular, that plasma was more likely to help than to do any harm. And the first patient was treated in March 24th, and here is some of the email traffic that preceded this. Jim Musser contacts me and says, I hope you're well. Realistically, do you think there's going to be a need for the disease? This is 2 weeks before. It's 3/14, so it's about 13 days before they actually use it. Texas moved first, and I got an email that they had treated them, and I will tell you that night I did not sleep. I kept wondering, oh my God, are we going to see some of these unusual side effects? And over the next few days, I was worried, and we saw a lot more patients being treated. Unfortunately, all the infusions were very well tolerated, and there was no evidence of antibody-dependent enhancement or anything like that. But you always have to be humble, and you always have to realize that when you do something to a human, there is always the possibility of harm. And certainly, as a physician, the most important thing that we can do is to do no harm. So the question became, is it safe? Well, by May, we had treated 5,000 patients. And I'm happy to report that there was no evidence of antibody-dependent enhancement. There was no evidence of cytokine storms. And then a couple of months later, we're up to 20,000, and these plasma appears to be safe. It's as safe as a unit of plasma. It's not like we're giving them antibodies and there is going to be a side effect. Today, the numbers are in the tens of thousands. And again, this holds. One of the things that is apparent is that physicians don't know how to use antibody therapies. Why? Because they have been abandoned in the 1940s. So people begin, the first use of antibody therapies is on people that are critically ill and are in the ICU and are unlikely to benefit. And you could see this in the dramatic reduction of mortality that we began to see. So in the first week of this program, 15% of all patients died. But 6 weeks later, the mortality is down to 5%. So all these patients have been treated. All these patients have had a benefit, presumably from plasma. But you're seeing that what happens with physician learning. Physicians relearned that they had to give it early. So then plasma usage moved out of the ICU into before patients became critically ill, and the results began to be that the responses were better. I will tell you, however, that antibody-dependent enhancement is still with us. This is a paper in Nature. You can see just being published online. And again, I reached out to my friends, Ann Arvin, Skip Burgeon, and we chatted about this. And they agreed that, it's a theoretical risk, but at least the human experience with plasma does not suggest that it's happening. It may be a concern with monoclonal antibodies, because there you have a lot of one type of antibody. We'll see. The trials have already begun, and again, people are watching very carefully. In the case of plasma, you have a zillion different antibodies that compose in a polyclonal response. So the likelihood that any one antibody will make a contribution to this is in fact, that is deleterious, it's very low. So is it working? That's what everybody wants to know. Well, you have the first randomized controlled trial in China. The Chinese obviously have to deal with this first. They set up a randomized controlled trial, but then they run out of patients. And even though they're not able to complete it, they show that if you use it before people get into the unit, that there is an effect comparable to remdesivir. That is, recovery is much more rapid. So in the meantime, Mount Sinai in New York, Methodist in Houston, China, Italy, Iraq, Iran began to report large reductions in mortality if given early before the ICU. These are not randomized controlled trials. These are basically trials or case series in which they compare those who got it to those who didn't get it. And you know what? There could be a lot of bias. There could be anything could come in and affect the results. But I would point out to you that the numbers of these keep going up and that they are in very different healthcare systems. Iran, Iraq, Italy, China, Texas, New York. And this is what happens. The Sinai experiences in Nature Medicine and Press, and you could see that if given earlier before people go into the unit, there's a big difference in mortality. And the Methodist Hospital is reporting the same results in mortality. That is, if people are given the plasma before they decompensate. This is a meta-analysis. We have a living meta-analysis in which we put together all the data that comes in. In fact, today we found another study that will be added to this. So the majority of the studies are small. But what you can see is that in every case, there is a reduction in mortality. Most of them don't reach statistical significance. But when you add them up and you do a formal meta-analysis, you basically begin to get real statistical significance in which you see a reduction in mortality. So this is a paper that was submitted last week, and this is the extended access protocol. So now you have thousands of people in the United States that have been treated. This was a protocol that was set up for safety, not efficacy. The question is, can you go back into it and find signatures of efficacy? And remember, everyone has been treated. So what happens, one of the things you can do is you can go back to the principles of antibody therapy. You could look what happens to the people who are treated early in the course of the disease versus late in the course of the disease. And in fact, the data shows that if plasma is given in the first 3 days, the mortality is a third lower than if you give it after 4 days. The same thing that Flexner finds in 1913. But most importantly, for establishing causality, is that we now see a dose response. These units were given without doctors knowing how much antibody was in them, but they save aliquots. And these aliquots are now analyzed retrospectively, and you can say, you can break it up into 3 categories, high, low, and medium. And then you could say, what is the mortality of the people that got high dose IgG to SARS coronavirus? Well, it's significantly lower than the ones who got low titer, even though presumably all of this had some benefit relative to ones who are not being treated. So here you see the temporal principle, here you see the specificity principle, and here you see the quantitative principle. We're basically rediscovering what people knew in the pre-antimicrobial era. It's important to know that convalescent plasma is highly heterogeneous product, even if you have high titer serum. Some of them have IgM, some of them have IgA. It's just every unit is different. And that is a very difficult problem for regulators for standardizing. How do antibodies work? Well, people forget that antibodies do a lot of things. They opsonize the virus, they activate complement. This is an envelope virus. They can mediate viral neutralization. They can prevent entry. They can mediate antibody-dependent cell toxicity. They can cross-link Fc receptors and affect immunomodulation. All these things may be happening, and some plasma may be better at one thing than the other. We have correlated neutralization with efficacy, but remember, correlation is not causation. It may just reflect that the more IgG you have, the more of these other functions happen. Convalescent sera is therapeutic in animal models of COVID. So in mice, if you give plasma from donors, people who recovered, you can see that you protect the mice, whereas you don't protect them with plasma from people who recover from MERS or SARS. And here, in the case of Osirium hamsters, this is not human. This is from hamsters, but again, you can establish an animal model for therapy. And there have been several of these provided additional support that this is working. So convalescent plasma has very different effects when used early or late. When people get COVID, they don't mount a significant response to between day 10 and 12. If you give it early, you're giving it to a host that had no antibody. So the antibodies are arriving, you're giving them immediate humoral immunity, and they're working with the cells that complement and everything is there. If you give it late, the effects clinically are much less, but you can still measure an antiviral effect. And one of the fascinating things is that people appear to be making their own antibody, but they don't clear the virus. But if you give them convalescent plasma, it clears the virus, suggesting that the antibodies made late are better than the ones that are made in the acute event. So the summary of the available data is that convalescent plasma reduces viral load. This has been shown by multiple studies, reduces inflammation, which in turn reduces respiratory demand, and all three combine to help the host translate into improved survival. So my opinion on efficacy, people ask me, what do you think? Well, there are strong precedents of efficacy based on historical use. We know a lot about antibodies action. This is not something in which you don't know the mechanism of action. You have encouraging signs from a variety of studies, and it depends when you ask. If you asked me in March when I wrote that editorial, I would have said maybe, but it's worth trying. By April, May, when the data from China and some of the data from other places was coming in, I would have said to you, possibly. By June, I was already aware of some of the data in the EAP. I would say probably. And today, I would probably say to you almost certainly. Now, what does almost mean? 70, 80, 90, 99% certainty? Well, I don't know where I am. I think it's very important as a scientist to always maintain doubt and to realize that things can be, you can be misled. I'm still hopeful for a good randomized controlled trial. And if a good randomized controlled trial was to show me that there was no efficacy as a scientist, I would have to abandon this view. But today, based on the totality of data, I think I would say to you that there is pretty good evidence that this is reducing mortality. We have two large randomized controlled trials at Hopkins. These are outpatient trials. They're funded by the Department of Defense. They've been led by Shmuel Shoham, that you may remember earlier wrote the protocol, and David Sullivan, supervised by Dan Henley. In some of them, we're asking the question, does antibody work in prophylaxis? If you have a big exposure, if you're sitting at home with somebody with COVID-19, does the plasma prevent you from getting ill? And the other one is an ambulatory protocol in which people who are sitting home with cough, fever, shortness of breath are being given the plasma. And the question is, do you prevent them from getting worse? Do you prevent them from going to the hospital? If you do that, then you could potentially reduce mortality tremendously and take a real bite out of the epidemic. So this is the epistemic climb to establishing efficacy. And I will tell you that this climb, this ladder, have been constructed in about 4 or 5 months. Here is the paper that we wrote back in the JCI. Then there are the antibody. There's a lot of work being done in antibody action, the neutralizing COVID. You have case series observations. Here is the first FDA action. Convalescent plasma is therapeutic in animal models. You have case series with matched controls from many places in the world. You have the extended access protocol analysis that I told you about, which incidentally is out there as a preprint. You can download it and read it on 35,000 patients. So we think that the FDA may move. I don't know. I don't have any inside information when that may happen, but we are hoping to get the gold standard, which are randomized controlled trials done. Two of them have been. One of the problems is that you cannot do them in search areas very well. So New York City, for example, search in March. They began to get their RTCs in place. It takes time to get IRB approval. It takes time to organize everything you got to do. And then guess what happened? They ran out of patients too. So fortunately, they continue to work at it. They're trying to find sites in the search areas, knowing full well how hard it is. When physicians are stretched, it is very hard to divert them to do a randomized controlled trial where everything is blinded to do so. But I will tell you that my colleagues are trying, and we hope to get to the point in which we can then say, for with some degree of definite, convalescent plasma is safe and effective. The final thought, though, is that convalescent plasma will always struggle. It is difficult to define pharmacologically. All units are different. There is no pharmaceutical company pushing for this. These are a collection of physicians and scientists that are trying to develop this. People prefer new stuff, even though it may be less effective. There is a bias how something so old can be so useful. The "we could do better" trap that you hear over and over again. But I think it will continue to be used against COVID-19, and it will be there for the next epidemic. And currently, it is the only therapy in which there is an association with a major reduction in mortality if used before the ICU, an association that we hope implies causation and that they will be proven to be causal. So I want to end with this note, because the audience is scientists, and scientists usually don't care too much about history. And I think that can be taken from Peter Medawars, immunologist, he got the Nobel in 1960 for immunological terms. The history of science bores more scientists did. And I have been a critic and a commentator about science together with Ferric Fan in many papers that we have written. And I'll just read you the last paragraph of this paper, in which we urge scientists to become more historians of their field. Science is more than a disembodied collection of facts. It is a uniquely human construct, a detailed and interconnected understanding of the natural world based on innumerable observations and contributions from individuals spanning thousands of years. History can help us keep science honest, with a keen sense of where it's been and where it's going. As Darwin observed, great is the power of steady misrepresentation, but the history of science shows how, fortunately, this power does not endure for long. At a time when facts have turned around, at a time in which a lot of the things get questioned, I think it is very important, no matter what field you are, to become very familiar into how the knowledge of your field was arrived at. How do you know what you know? And that, if you do that, that will give you, I believe, an advantage as a scientist. Thank you.
Thank you, Arturo. That was a fantastic presentation, and I can say a heroic effort from your entire team.
I'm going to stop sharing the screen. Okay.
All right. So the audience is curious. Questions are coming in, so we'll dive right into the questions. We have time, so we're going to let the audience ask their own questions. So Eric, please unmute yourself and ask your question.
So, hi, Arturo.
Hey, Eric.
Nice to be at Einstein as well. So I really appreciate your message about the history of science, and I encourage all of my cell biology students to always think about this as well. But one thing that you mentioned, and I've been curious about for a long time, is that this approach of using serum has been applied for hepatitis A, in which people who have been potentially exposed get gamma globulin therapy. Now, if I'm not mistaken, that's not actually immunoglobulins raised specifically against hepatitis A. Is that correct?
Oh, that's because in some parts of the world, most people get hepatitis.
So it's just commonly
Exactly. So if you make a gamma globulin preparation of a population that has a high prevalence of hepatitis A, you have essentially an antibody therapy or an antibody prophylaxis. It used to be in this country that they will give you immune globulin when you travel to those regions as an IM shot. That is no longer so much used, but that is correct.
I see. Okay, thank you. That helps clear that up.
Arturo, yeah, that was heroic what you guys got done. I wanted to toss in a question about what do you think about the other components of plasma, cytokines,
Right.
complement.
Exactly. So when you give a patient a unit of plasma, you're giving them 200 cc's total. The total volume of blood is 5 to 6 liters. So it's a very, very small amount. So it's unlikely that the amount of complement or the amount of coagulation is going to make much difference given the amount of it. But a fair question is, what about exosomes? What about cytokines? Well, they may very well be there, but when you get a dose response with antibody, that begins to associate the active component as antibody. So the other stuff may be there, maybe it's doing some good, but it is the antibody that appears to be associated with it.
Yeah, and on that note, did you guys titrate IgM and IgA in those things as well?
No. This is, Lauren, this has been done emergently. The samples are there, and there will be a lot of analysis for years to come. It'd be great to see the people who really responded to try to understand the epitopes that were recognized in the plasma that was given. All that is in the future. Right now, we are trying to get almost like the 30,000 foot view to try to determine how to try to use this and how best to, with the fine analysis to come later.
Yep, well taken. Charles, can you unmute yourself?
Hello. Hi, Arturo, thank you so much for that talk. I think it takes a lot of guts to, think the way how you've been thinking about this problem and trying something unconventional, especially in the face of a global pandemic that people really haven't seen in the past 100 years. I have two questions. The first is you were talking about the time dependence of when the convalescent plasma is administered to patients. And it seemed like between maybe day 3 and 4, you can see almost precipitous drop off in the efficacy of the plasma. And, for a virus like COVID-19 that seems to have a very long incubation period, I was wondering if you could talk a little bit more about some of the challenges of specifically, I guess, administering or identifying, what's the best way to give the drug or the plasma. And the second question I had was in the beginning when you were kind of giving us the historical overview of this field, you had mentioned that there are some pathogens that seem to be readily addressed with this strategy, and then harder ones like like tuberculosis, that's a lot more difficult. And I was wondering if you could identify for us what maybe some of the general principles are that would I'm
happy to, because that's what I've done. That's been my research work most of my life.
Okay.
So let's go back to your first question. Why does antibody work best when given early? I think that is a fundamental immunological question. There are many ways to think about it. One of them is obviously the inoculum is smaller early on, and you're given a set amount of antibody, and at some level you got to have stoichiometry of reacting with. The other possibility is that even when antibody, even the pathologic process that once you have that inflammation basically driving the damage in the lung, even if you neutralize the virus that is accessible to antibody, you're really not going to be able to change the outcome of disease. So you're right. I think there is a narrow window. I think that once what we have learned is that even in a few months, physicians can be taught to use it early. Today, I think it should be used, and my view is that anybody who gets admitted to a hospital, the likelihood of dying from COVID is many orders of magnitude greater than the side effect of plasma. Therefore, probably should be used very early on anybody who gets admitted. Your second question is what I worked on all my life, and that is, why is it easy to make protective humoral responses to some microbes and not others? And what I can tell you is that for things like toxins, where you have an epitope and you can just make a neutralizing antibody, that's relatively easy. For pneumococcus, where you have a capsule, and if you can drive it into a macrophage and put it in the right phagosome and you can get it killed, that is still understandable. The problem is that for the intracellular pathogens, TB, Salmonella, Francisella, these organisms have very, very complex surfaces. And you can make protective and non-protective antibodies. And in the case of the fungi, which is what I basically worked a lot of this out, is you tend to make both types at the same time. And the net result is that they neutralize each other. So one of the things that we had to learn was how to make a protective humoral response. And over the years, we showed that you could make protective antibodies to TB, to the fungi and all that. But the problem is that the complexity of the humoral response often resulted in what appeared to be no efficacy. And immunology made a logical error. And the logical error went like this. They could protect against some pathogens with antibodies, and they put them into the humoral immunity category. And they could not protect against those, and they put them into the cellular immunity category. And they made a logical error that goes back to Aristotle, which is they used the absence of evidence for its absence. And that drove a lot of immunological thinking in the 20th century. And the revisions of now that you can probably protect against any pathogen with both cell-mediated immunity or antibody immunity if you just happen to hit the right stuff. But it is an example where the training of scientists is often not adequate for the kind of challenges, logical challenges that they encounter.
Thank you very much.
All right. Arturo, I have a question. We spoke about the efficacy of plasma for the treatment of SARS-CoV-2. What are your thoughts on the prophylaxis? I mean, can this be used for, say, first responders or ER doctors, or even somebody who's had an accidental exposure? Do you think that this approach would work?
Yes. From 130 years of working with antibody, the expected result is that if given prophylactically, it will work much better than if given therapeutically. So you can imagine, for example, perhaps not with plasma, but once we have gamma globulins or monoclonal antibodies with long half-life, that you could give everybody a shot with a half-life of 20 days and provide long-term protection, especially people who are going to likely have a high likelihood of being exposed until there is a vaccine. I'm very optimistic that we're going to have a vaccine. I'm very optimistic seeing what's in the plasma, seeing how high titers of neutralizing antibodies that we are going to basically beat this thing. But I would agree with you that plasma could be used to protect people. Plasma can be used early. Hopefully better antibody preparations are underway. And when they are, that we can provide people with immediate immunity.
Is that being tried?
Yes. Well, it's been done in 2 randomized controlled trials. Here in Hopkins, we're using plasma in first responders that had a major exposure, in people that are home taking care of somebody with COVID-19. And those are randomized trials. Those you get either, nobody knows what people are getting because the units are all barcoded. The only one who knows is the data safety monitoring board. So one either gets normal plasma to get at Lauren's comment about all the goodies in plasma, or you get convalescent plasma. And we'll see. The expected result is a convalescent plasma will have a big effect.
Thank you.
To be clear, with my first question, I did mean convalescent plasma, just that, the levels of the different goodies would presumably change in response to the infection.
Right. No, although, we're taking it from people that are already well, but you're right, there could be still cytokines around.
Yeah.
Exosomes, who knows? Yeah.
I think there will be long-lasting signals.
Yeah.
I think. I had a follow-up question about antibodies. When you titrated the IgG, that was just IgG. That wasn't neutralizing antibody, right?
Oh God, you're getting us into the last 2 months. The experiments were done with neutralizing all was done neutralizing antibody too. But in negotiations with our sponsors, which is the government, it was decided for the preprint to report only total IgG because currently that is much easier to measure.
Exactly.
Right.
So,
but the data, if we have the data with neutralizing antibody, and you could just do the same analysis.
okay, because the reports I've been seeing, I've been seeing like 3 to 5% of IgGs from sera are neutralizing for SARS-2. Are you guys seeing a lot more than that?
Well, I mean, one of the things that is very clear is that the titers peak around a month to two, and then they rapidly decline. So most of this plasma that was used from people who had the disease relatively recently, but there were quite a few units that had very little antibody, very little antibody, and there were a lot of people that seemed to be clearing the disease with high titers of IgG that's specific for very little neutralizing antibody, and some people appear to be clearing it without any significant antibody response, suggesting that whatever is happening locally.
Yeah, I mean, I was just, since, I think we were all worried about antibody-dependent enhancement earlier. I'm starting to be much less worried about that. So, the non-neutralizing antibodies through all those other pathways should give you a good boost as well.
I was, even though I was confident, I was worried too. I think you have to be, especially when you're dealing with people's lives.
Yeah.
But then again, it was history suggested that it was going to be okay, and it turned out to be okay.
Mm-hmm. Yeah. I've got a question. I'm fascinated about how the whole convalescent plasma, the logistics of it work. So, for instance, you say each bag is barcoded. So how many patients would be combined for?
They don't get combined. That's one of the beauties.
Okay.
If a patient who gets COVID produces 2 to 3 units.
Okay.
So one person can help 2 to 3, and very large people can produce as many as 4. They are never combined.
Okay.
They are treated as individual units.
So then the obvious question is, do you guys, when do you think you'll have enough data to power a follow-up study on what are the properties of patients that produced very good plasma versus those that produced?
So I think right now, by the way, the history of this and the preprint is that what happened is this was never set up for efficacy. What happened is the FDA, once you began to said, can you look for signals of efficacy?
Yeah.
And they have paid for a lot of this analysis, FDA and BARDA. I think right now the major goal is to see if you can define a useful unit.
Yeah. With
IgG. I think as we go forward, you want to know whether IgM, there's evidence that IgM is really important in viral diseases. IgM matters, IgA matters, as well as looking for epitope mapping, taking all these things and express all the pieces of the spike protein and see whether there's a correlation between some epitope. But that's all is in future, I think.
Yeah.
I think right now we really have to try to get the clinical data tied in as best we can with simple immunology.
Yeah. But
that is exportable. I mean,
Yeah, but on the team, you guys must, have put together that, like, let's say Susan and Timmy, produced this killer plasma that,
Yep.
like you call them every 10 days and they're like, we need you back here for
Yep.
another
Yep.
donation.
I mean, so it looks like plasma. Plasma is going to come into platinum, platinum, gold, silver, and bronze.
Mhm.
And I think that's going to be the case. And one of the concerns, by the way, of regulators is that if an approval is issued, there is going to be a tremendous demand for plasma, and we need to be able to identify what is the useful unit.
Mhm.
When you know very well that every unit is different, and the question is, should you give 1 unit or 2 units? There are a lot of people who have received 2 units, and if you get 2 units, well, you're hedging your bets, right? Because for all you know, the effects are coming from ADCC. They're not coming from neutralizing it.
Yeah.
So,
So why wouldn't you want to combine in maybe, I mean, again, these N are really hard to come by, but if you had an arm of your study where you combined, maybe it is some
so there is whenever you use plasma, there is always a very small risk of infectious disease, right? Because even with HIV screening, hepatitis screening, all the other screening, the moment you pull stuff, then you create the risk. 1 unit can cause a risk to the entire lot.
Fair enough.
And I think that's safety. You want to keep things separate, even though you, again, are going to be given, everybody's going to get a different product.
Yep. Nope. You're right. Sarada, did you have another one?
Yeah, I just had I guess we touched upon this. I mean, my question or other comment was on the batch to batch variability, which is, I guess, what we discussed right now. So how big do you think is this variability going to be an issue? I guess the batch to batch variability between plasma.
So I think I can tell you it's an issue already, because you can find in the database that people who got the highest amount of antibody had the lowest mortality, even on a database where everybody was treated. I think that what we need to do is we need to really mobilize donors and not worry too much about the variability, but try to identify really good unit of plasma, and then take the rest of it and ship it off to make gamma globulin. Because even low titer plasma is useful for making gamma globulin. In gamma globulin, they do what Lauren suggested. They take 10,000 units, they mix them together, and then just pull out the IgG and they concentrate it. So you have a concentrated IgG product with high titer, that is, every vial is the same, at least per lot, that can be used. So the bottom line is we need to encourage everyone to donate. And the great donors out there, that can be used directly into patients, and the stuff that is so can go into producing other antibody products.
This is also fascinating. How long can you milk someone?
What a great term, milk someone. Well, you can generally bleed them every 2 weeks. So every 2 weeks, you can, the standard practice is every 2 weeks. You'll probably do more than that, because you're just removing plasma, which is albumin and antibody, and those things get remade very rapidly. But I think cautious transfusion practice is you bring them in every 2 weeks. If they produce 2 to 3 units, in 2 months, one person may be able to help 10 people. And that's a really good ratio.
That's amazing.
And also you can freeze it. You can freeze this stuff.
Yeah.
If you generally in the United States, we freeze it for 1 year.
Hmm.
But if you freeze it at much lower temperatures, you can freeze it for many, many years. Antibody stable. I have antibody that I made 25 years ago and it works. And antibody molecules are quite rugged.
Yeah. Yeah. My last question is, what would help you guys? Like what, I mean, other than getting the disease and having a nice neutralizing antibody response and becoming a milk cow, like how else can we?
I really appreciate it. I think that even today the whole thing is a struggle. It's a struggle. For example, we're getting criticized. People are saying, "How could you treat so many people and not do a randomized controlled trial?" And the answer is that people are looking at it and putting these two things in opposition. The extended use protocol was put in compassionate use. The physicians decided that this was the only thing they had, so they used it a lot. The problem that the randomized controlled trials are not completed is not because of the extended use protocol, but rather because they were put in place and they're in places that were not patients. So one way in which you can help is you can actually become educated a little bit into this, and when these discussions come on, like not repeat some of the things that are said over and over again, that in fact don't create straw people in that way. I think another way that it can help obviously is to get involved, donate. This was a completely grassroots effort. There was no government coordination. There was no industry coordination. This was put up by people working together, thousands of people across the country that did this.
Mm-hmm. And
it's an example that people can get organized to do so. And it may be one of the few things potentially that as a nation, the United States has led in this area, where has not led in some of the others.
Yeah, not lately. I mean, so if you guys had more funding, I guess you'd be able to scale up your efforts to other sites and et cetera.
So we have been, for the randomized controlled trials, we have received generous funding from the Department of Defense. The problem is scaling up. Think about bringing in a new site. You need to identify a PI. You need to identify a place that you can bring somebody who's infected. You need to transfuse it. You need to get the units out there. You need to go through their IRB. It's even in the conditions of a pandemic where people are working really hard to set up and to scale these things up rapidly. It's very hard. Yesterday, the New York Times told you that the monoclonal antibody trials are in fact running into the same problem. It is hard to do clinical work, period. You don't control the variables. You're dealing with humans, you're dealing with people, you're dealing with regulations. And that's what is needed. I mean, it's needed more and more people to be in the front lines willing to do this.
Yeah. Arturo, I'm in awe of what you guys managed to put together in just a few months. I mean, that's...
it was an effort of thousands, and people pretty much dropped what they were doing and they did it. And today we are now trying to get at the efficacy question and we can step back and say, today in the United States, if you get COVID-19 in most places, you have access to plasma. That's a success story. Now the question is, can we figure out how to make it better? Can we apply some science to it to really sort out, the nuances in the therapy? And to do it, because I think we're going to be using plasma into 2021. Mm-hmm. The monoclonal is in 2 weeks in September. The monoclonal are not going to be available to flow for most people in the fall. So plasma remains the major source of antibody therapy into 2021. So let's try to figure out how to make it work better.
Yeah. And when even when the monoclonals are available, there's a good chance that, we'll pay like $15,000 for them.
Right. It's not going to be $300 per dose. And the other thing is that hopefully they can scale up capacity. I mean, something like this, no monoclonal has been produced for the amounts of treating the people that need to be treated. And also in underserved areas of the world, this is the only thing you're going to have. You're not going to have in areas that are under-resourced, You're not going to have the latest antivirals, and you're not going to have monoclonals, but you're going to have survivors.
Mm-hmm.
And you can have potentially these therapies can be developed locally. This is a therapy that can be developed at the level of a hospital. It doesn't require a pharmaceutical infrastructure.
Have you had people reach out to you from very under-resourced places?
Oh yeah, I mean, I am totally, because I'm a Spanish speaker, we spend a lot of time in March and April with South America, and South America is using it, and they move rapidly to use it. Some countries move much more rapidly than others, but you know, you're talking about countries that have only a few ICU beds, but they can do transfusions and they can harvest plasma. So they can potentially make this therapy locally. And what we need to do is we need to define the parameters by which this works, and then just put out the information and they'll be able to use it.
Yep. We've got another audience question. Janet, can you please raise your hand? Okay, can you unmute yourself? And
Yeah, I'm on mute now. I'm on mute.
ask
Can you hear me?
away.
Not going through?
Yeah, I can hear you, Janet.
Oh, yeah. Just wondering how the plasma treatments will work with dexamethasone and remdesivir.
So we have three therapies right now: convalescent plasma, remdesivir, and dexamethasone. Dexamethasone only has been shown to work on people that are really, really ill in the ICU. And there it drops mortality by about 20% because it probably reduces inflammation, reduces lung damage, and people can get off the respirator. Remdesivir has been shown to reduce symptoms, has not been shown to reduce mortality. It may very well be that in the future, the clinical trials will show it, but it hasn't been shown to it. Plasma is the only therapy that is associated. Notice that I'm using the word associated. I don't want to imply causality yet. That is associated with a reduction in mortality if used early in the course of disease. So I think that these are very different therapies. One is an antiviral, the other one is an antibody, and the other one is a steroid that have different roles at different places in the course of disease.
Okay.
And you know, one of the things is that we should be very proud of the scientific establishment. You know, in March 1st, the only thing we had to give people was oxygen. And the only difference between what we had in March 1st in 1918 was that we had respirators. In 4 or 5 months, we have more things that we can offer. And this has been the result of the medical scientific establishment that is working in overdrive to do this. And I'm very, very proud of my scientific colleagues on how rapidly they responded to this calamity.
Thank you.
Yeah, Arturo, we'll let you go. Thank you so much for your time.
I'm afraid to open my inbox since I haven't looked at it. I haven't looked at it in an hour and a half.
But that was amazing science and amazing history lesson.
Thank you. And thank you for being such a wonderful audience. And thank you for inviting me.
Thank you.
Take care. Bye-bye.
All right, Arturo, be well.